guys, and welcome to Lab 4 on cellular observations, as seen here in your lab manual. Now, there is a lot to be said about the text in this lab manual. So your lab manual goes through and discusses the concepts of cell theory, being that all living things are composed of cells. Your lab manual goes through and discusses how all cells arise from pre-existing cells, i.e. that life only arises from other living things, and third tenet here that the biochemical reactions upon which life is based occur within cells. So cells are the structural and functional unit of life. Now that's all well and good, not something I'm really focusing on in lab. That's more like our lecture material that we've discussed previously. Now they also go through and have a big conversation on the diversity of life. Now, what they're really doing is they're trying to explain to you the concept of Linnean Taxonomy or Linnean classification. Carolus Linnaeus was a naturalist a long time ago. Let's see if we've got a date, I believe in the late 1700s. But regardless, he came up with a method of outlining organisms in a fashion that would make them sort of easy to figure out who fits where in terms of relation. And the way this falls out from Linnaeus's capacity was from kingdom to phylum to class, order to family to genus to species. And this should look very familiar with you from Biology 1. Now, as we well know today, there are also domains, right? So there's a little more to say here, but this Linnean system of taxonomy is still very important, and it wouldn't be the worst idea for you to go ahead and understand that there are domains, kingdom, phylum, class, order, family, genus, and species. You're talking about identifying organisms based off of similar physical structures and or relationships. Okay, so this is the way we line things out, and they go through in a little while and talk about the concept of binomial nomenclature, which I went ahead and thrown up here for you. Binomial nomenclature, which is on page 53, as the way in which we write species names down. This is very important. You will be doing this for me again. Pay attention. The way this works is as follows. Anytime you're writing out like a species name of anything, like green swordtail being Xiphophorus helleri, or a Florida panther being Pantheraleo, Felis concolor, the lion's Pantheraleo. Felis concolor is a Florida panther. Anyway, anytime you're doing this, it's a two-name system, like Felis concolor, or Pantheraleo, or Homo sapiens, or Cambarus diogenes, okay? All of these, it's a two-name system. It's binomial nomenclature. The name is two parts. Binomial, two, nomenclature, name. It's a two-part name. And the way that we do this is that we call it a genus and species name. You are a Homo sapien. That is your genus and species. Lions are Pantheraleo. That is their genus and species. Procambarus acutus is a genus and species. It's binomial nomenclature. And one more little tidbit about this before I move any further is we always write this out in a very unique fashion. And that is that the first letter is always capitalized in the genus, and then in the species name it is always lowercase. And further, if you were handwriting this, you would typically italicize it. As you can see, these are italic. You can underline them. And all of these are acceptable ways of doing business. But regardless, this is a Linnean system of taxonomy. As we go through, we're going to be looking at some species names or general organismal names. And it's all really based off of the work of Carolus Linnaeus way on back in the day, figuring out a way to classify organisms. Yeah, that works. That works just fine. Now, also in here on page 52, there's a brief conversation about autotrophs and heterotrophs. This is worthy from my perspective. You should already know this, so this is not rocket science. The idea is as follows. There are autotrophs. Autotrophs are organisms that survive off of the sun's light. They are photosynthetic. Autotrophs conduct photosynthesis using chloroplasts. These are the organelles within the cell, and I think that's good enough. Let's not just get too deep into this. But regardless, autotrophs are like plants, okay? They have chloroplast in their tissues, which allow them to conduct a process called photosynthesis. Yeah, that's enough. And then, of course, there are heterotrophs, like you and I are heterotrophs. This deer is a heterotroph. What we do is we conduct cellular respiration, And cellular respiration is conducted via mitochondria. So you will find mitochondria in both plant and animal cells. You will only find chloroplast in plant cells for our purposes. Okay, for our purposes. We are hot heterotrophs. That means that we accrue energy secondarily, so we eat primary producers, and we make our energy via cellular respiration. It's good enough for me. Good enough for me, folks. And next, on page 52, there is a little bit of a conversation on prokaryotic and eukaryotic cells, which is also worthy of our considerations here in lab. That is to say that prokaryotic cells are very small compared to red blood cells. All right, prokaryotic cells are very small compared to red blood cells. Or let me rephrase: prokaryotic cells are very small compared to eukaryotic cells. Prokaryotic cells lack membrane-bound organelles. Prokaryotic cells have their DNA in a single ring, kind of just hanging out in the cell in a random location, whereas eukaryotic cells are very much larger, orders of magnitude larger, and way, way more complex. They have membrane-bound organelles. Eukaryotic cells have nuclei with nucleoli. They have all sorts of other structures which allow these cells to be much larger and much more efficient in the way that they do business. Perfect. And that takes us to domain bacteria. We're going to look at prokaryotic organisms. So in your lab manual, they go through and they talk about the various domains being bacteria, archaea, and eukarya. Eukarya, English. Bacteria being the organisms that are ubiquitous with the world around you. They're just everywhere. Okay, in your gut, on your skin, on you right now, you have bacteria. There are archaeans, which are the extremophiles, only found in like sulfur ponds and deep sea vents. And then the eukaryotic cells in domain eukarya, which you should know a little bit more about. In fact, on page 53, there's this table. And you'll find this table later on in the lecture as well. But this table is your friend. You need to know about the various kingdoms, the way they are sort of set up cell-wise, the way they acquire nutrition, and the way they are modal, the way they move. All right. So first and foremost of these, just flip my page here a little bit. We have a variety of organisms that we're supposed to look at in lab this week. Okay. Now, full disclosure: if you flip your page a few times and you get to the back of page 69, so page 69. And page 70, there are some pretty good images of this. I went through and found some better ones and lined them up in a fashion that would make more sense to you. So if I were you, I would be on page 59, and I would be making notes about these organisms as I talk about them. On your lab test, I will ask you to identify these organisms based off of the way that they look, and then to tell me a little bit about them. Like, how do they move? Or how do they conduct their metabolism? That sort of thing. So let's go through and look at it. All right, on page 59, it asks you to look at Anabaena, Oscillatoria, and E. coli. These are in domain bacteria, which makes all three of these prokaryotic. So Anabaena, as seen here, both of these, Anabaena and Oscillatoria, these are blue-green algae. It's the way that we describe them. They're bacteria, but they're blue-green algae, so they are photosynthetic. They will have the capacity to use sunlight for general photosynthesis. And boy, they're kind of found all over. You know, they were in the pond water samples that I pulled previously. Like these things are kind of everywhere, and they're pretty large. You can see them pretty clearly, even through the microscope, a light microscope. Whereas bacteria like E. coli, these are very small. You can see the magnification level is quite different here. And E. coli are your classic intestinal bacteria, gut content. They hang out in there and break down certain nutrient molecules and basically keep worse stuff from getting in. So E. coli are all over our systems. They are all within our intestinal tract, and these are considered bacillus. They have little rod shapes. Okay, each individual little one here, this is an individual cell, and you can fit 100 of them inside of one of these oscillatoria cells. They call these oscillatoria because they oscillate to orient themselves to face sunlight more effectively. Yeah, so Anabaena, Oscillatoria, and E. coli. You need to be able to look at it and tell me what it is, tell me that it's prokaryotic. For these, tell me they are photosynthetic. Maybe for those, tell me they're gut bacteria. Yeah, that was pretty well. Next, domain Eukarya, kingdom Protista. And here's that chart I was telling you about previously, so we're going to start on the Protists. Protists are a catch-all. Protista contains organisms that are very distinctly different from one another. I mean, there's a whole host of very unique organisms. Some of them act like bacteria, some of them act like animals, some of them are small, some of them are large. It's just highly varied. Basically, Kingdom Protista is the catch-all for everything that doesn't fit somewhere else. So let's talk about it. This is amoeba, specifically amoeba Protists. These are pretty cool single-celled organisms. So you can see their nuclei inside of there, and what they have are called pseudopods. All right, they can exude their cytoplasm and their cell membranes, and sort of hold on and then crawl and then crawl. They have these pseudopods and it allows them to crawl. It's like the blob, man. So that they move using these pseudopods to kind of crawl along slowly using cytoplasmic streaming. Really cool, man. Super cool. They have a whole host of different life history strategies. They're very fascinating. Next is Euglena. Now, Euglena is interesting because they bear flagella. Okay, Euglena are also single cell. You can see the nucleus here. If you look on this image, you can see its flagella sticking out. So it's going to beat that flagella to allow it to swim from one place to the next. So these are a flagellated protist. Okay, Euglena, a flagellated protist. And that's my son out there. He's having a great time. And then compared to that is Paramecia. Okay, Paramecium are ciliated. So what I'm really trying to do here is I'm showing you three different forms of locomotion. There is pseudopodal motion, there is flagellated motion, and then there is ciliated motion. If you look at the border of this paramecium, it looks like it's got a little fuzz around it. And what those are cilia. They're like little hairs. You saw these in the previous lab video where we did the pond water. You can see the organisms beating their beating their cilia to move from place to place. That is classic paramecium. Well, a paramecium displays the same form of locomotion. How about that? So those are very interesting organisms. Very interesting indeed. Pseudopods, flagellum, cilia. The question would look like this. Identify this organism. It's amoeba. Question 2, is it prokaryotic or eukaryotic? It's eukaryotic. What kingdom is it in? Animal? No. Protista. There we go. And what's its form of locomotion? Locomotion. It has pseudopods. That's what I want out of this. All right, you need to be able to identify and tell me about it. All right, next is fungi. Kingdom fungi. So in this, there are a few concepts that I really want to get across to you. Fungi are not solid structures. So when you see a mushroom outside and it looks like, you got your cap at the top and the stalk down below, or you go to Publix and you buy some, nice portobellos or something like that, that is not a solid structure per se. If you look at it under magnification, it's actually made of a bunch of individual strings. Fungi are just a big conglomeration, if you will, of individual strands, growing strings, and they will wrap around each other in unique ways and form a mushroom or form a ball or do whatever they need to do. These strings are called the hyphae. Okay? Fungus is made of what's called hyphae, and these are the little string-like structures that give rise to other parts of the fungus. And again, fungi is super diverse, super diverse. There's all sorts of different types. The one that we were to look at in lab is called Rhizopus. Now, Rhizopus is kind of interesting. This is a fungus that's typically found just on the forest floor around us. It can get into plants, it can get into animals, it can infect you under the right circumstances. But you probably are most aware of it as bread mold. Okay, it's oftentimes seen as bread mold. Now, Rhizopus has two unique structures that you need to be aware of. These are its sexual stage, referred to as zygospore formation. These are all zygospores. And the second is an asexual reproductive stage, referred to as a sporangium. The concept is as follows. Under a harsh environment, when life isn't as good as it could be, my kids, man. when life isn't quite as good as it could be, you want to have sexual reproduction so that you can have more diverse genetics, because the more diverse your genetics, the better your chance of survival. But if life is easy and smooth and everything's perfect in the world and you're just doing your thing and everything's wonderful, then you want to reproduce asexually to make as many copies that are identical to yourself as possible, because that will be the perfect organism for the situation you find yourself in. So, how do you know the difference? Zygospore formation is when you have two hyphae grow together and touch. Okay, that's two individual hyphae. They grew together, they touched, and they mated basically and produced sexually produced spores. And this is a spore sac. It's forming, and it's going to pop and release sexually produced spores all over the place. Okay, so the two hyphae coming together, ball in the middle. That's classic zygospore. That is sexual reproductive stage. Compared to a sporangium. So what you have here is the hyphae is growing, and then it stops, and it forms a spore sac, fungal spore sac. This is asexual. It's not touching any other hyphae. That's going to rupture and release spores all over the place, and they're going to develop new hyphae that are going to spread just like the other one did. So this is classic fungus, all right? Classic fungal hyphae. All right, next is domain Eukarya, kingdom Animalia, and kingdom Plantae. So animals versus plants. These are human cheek cells. I don't know if these might be mine. I honestly forget. But in this, it's pretty obvious to see. These are always stained using a stain called methylene blue. So when you see blue cells with their regular shapes, they're typically human cheek cells, good old-fashioned animal cells. Your dogs look exactly the same. And in these, you can see their nucleus. This would be a cell filled with cytoplasm. You can see the cell membrane here. These are kind of broken up because they're scratched off the wall of the mouth. But these are classic animal cells, right? Classic animal cells. If I were to pan back to here, that cell right there, in its entirety, exactly the way it looks, would be represented inside of this cell. Okay, it's all there, all the same stuff, visible to you through a good light microscope at 400-time magnification. That is compared to plant cells. Now, plant cells, you can see, are very, very different. These cells will be, and this is so important, these cells will be quite square. All right, the cells are indeed quite square, and these square cells are so because they have cellulose-based cell walls. Cellulose-based cell walls give them this square appearance, and interiorly you can see the chloroplast. These are all chloroplasts, little chloroplasts all over the place that would be conducting photosynthesis using the sun's light to make sugars in essence. Perfect. Perfect. Let's make sure we got everything we need here. Yeah. Yeah. I think I'm pretty happy with that, folks. Yeah, good. All right. Now, a quick couple of comments further. In your lab book, there was previously a couple of models of plant and animal cells. It looks something like this. In other words, something like these. All right. You are responsible for basically knowing these two models, a plant cell versus an animal cell, and being able to identify the parts and pieces therein. It's no different from what we were just talking about. So you can go to page 55, and you can see a breakdown of these models. But I went through and put green arrows on the bits I thought were most important, but all of it, but green arrows in particular. So I'm just going to run through these with you real fast. This is obviously an animal cell. You can tell that because there are no chloroplasts, and it's round with no cell wall. So you can see the cell membrane here. You can see the mitochondria, which would conduct cellular respiration, right, making ATP. You can see the Golgi body there, which would be for packaging. You can see the rough and smooth ER here. It's kind of hard to tell which one's which, but these are either rough or smooth ER. Endoplasmic reticulum, all the same. You have your nucleus, which would house chromatin. You have your nucleolus, which is the site of ribosome formation. Perfect. And then by comparison, we have our plant cell. Now this is obviously a plant cell because you can see the cell wall surrounding it. Also, you can see a big old honking central vacuole. Central vacuoles are classic plant cell indicators, if you will. You still see mitochondria, which would still produce, or would still do cellular respiration, but this also contains chloroplast. and chloroplasts conduct photosynthesis. And yeah, nucleus, nucleus, it's all there. So that is really all you needed for this fourth lab. Like normally this would take forever because you got to do a lot of microscope work, but because we're not in lab, we aren't. We're just not. So what I'm going to try to do is find a little virtual lab and see if I can find a way to make you answer some questions on it. But other than that, it's good enough for me. All right, thanks guys.